The air is largely composed of nitrogen at 79%. Oxygen follows at 21%, and the amount of carbon dioxide, water, pollutants, and other gases is a negligible amount of less than 0.1%. These gas molecules exert a pressure around us. The amount of pressure exerted by a molecule is the same regardless of the type of molecule it was. In our current environment of 79% nitrogen and 21% oxygen, nitrogen exerts a pressure of 600 millimeters of mercury upon us, while oxygen exerts a pressure of 160 millimeters of mercury. If our environmental conditions changed, as they do in situation 1, and we maintained pressure but saw an increase in nitrogen and a decrease in oxygen, you see that there is only a change in the amount of pressure exerted by each molecule. Again, the amount of pressure exerted by a molecule is the same regardless of the type of molecule it is. On Earth, we do not see large changes in the amount of nitrogen and oxygen in the air. What does change is the amount of air pressure exerted upon us. As we move to higher elevations, the air becomes thinner and pressure decreases. In situation 2, we had a pressure drop from 760 millimeters of mercury to 680. If we look at the amount of pressure exerted by each gas, we see a proportionate decrease in pressure of nitrogen and oxygen. Both gases decrease the amount of pressure they exert upon us because of the overall decrease in pressure. We will see the effect and impact that changes in pressure would have on step 2 of our respiratory exchange. Gas exchange at the alveoli is ideal. The alveoli cells are incredibly thin and very closely arranged in relation to pulmonary capillaries. Reconditioning of blood is based on diffusion down its concentration gradients. Since this is diffusion, it is a non-energy consuming movement of oxygen and carbon dioxide. The air brought into the alveoli is low in carbon dioxide and high in oxygen. The blood moving through the pulmonary capillaries have just returned from the systemic circulation. It is low in oxygen and high in carbon dioxide because oxygen was released to the tissue and carbon dioxide was brought into the bloodstream. Because diffusion moves down its concentration gradients, we are going to see oxygen move from high concentration to low concentration. It will diffuse from the alveoli into the interstitial fluid and into the pulmonary capillaries. Carbon dioxide is going to move in the opposite direction. It will move from the plasma and into the alveoli. We can determine the actual pressure gradients of oxygen and carbon dioxide in the alveolar sacs and the air because we know atmospheric pressure and the relative percentage of each gas. Oxygen in the air has a partial pressure of 160 millimeters of mercury. The partial pressure of carbon dioxide is about 0 millimeters of mercury. As the air moves through the anatomical dead space, water is added to the air molecules. Also, remember that 150 milliliters of air is old air. Therefore, by the time the oxygen reaches the alveolar sacs, it only has a partial pressure of approximately 100 millimeters of mercury. This partial pressure is relatively stable between breaths. In the alveolar sacs, the partial pressure of carbon dioxide has increased to about 40 The partial pressures of these two gases ensure that oxygen is going to be brought into the lungs because it will move down its pressure gradient. Carbon dioxide in the alveolar sacs is greater than the partial pressure of carbon dioxide in the air. This will force the carbon dioxide to move out of the lungs and into the air. The maintaining of these pressure gradients is vital for ATP production. The blood returning from the systemic system with a partial pressure of oxygen of approximately 40 millimeters of mercury during resting conditions. It also has a partial pressure of carbon dioxide of 46. We will diffuse from high to low. We will see the following occur at the alveoli. Because the partial pressure of oxygen is higher in the alveoli, we will see oxygen diffuse from the alveoli into the interstitial fluid and then into the plasma. We will see carbon dioxide move in the opposite direction because the 46 millimeters of mercury pressure is greater than that in the alveoli. Blood leaving the pulmonary capillaries will always leave at equilibrium with the alveolar sacs. In this example, the blood leaving the pulmonary capillaries will have a partial pressure of oxygen of 100 millimeters of mercury and a partial pressure of carbon dioxide of 40 millimeters of mercury. The ability to leave at equilibrium is vital to the body's ability to meet energy demands. In the example displayed, we see that the body has become increasingly metabolically active as the blood returning to the pulmonary capillaries has a partial pressure of oxygen of only 25 millimeters of mercury, and the partial pressure of carbon dioxide has increased to 55 millimeters of mercury. Diffusion rate in this case is going to increase because the concentration difference has also increased. Even though the partial pressures have dropped, the blood will still leave the pulmonary capillaries matching those of the alveolar partial pressures. The blood's need to reach equilibrium also explains why working out at higher altitudes places more stress and is more difficult on the body. In this example, we see that the partial pressure of oxygen in the air has dropped from 160 millimeters of mercury to 130. This would correspond to a pressure drop in the alveolar sacs from 100 millimeters of mercury partial pressure of oxygen to 70 millimeters of mercury. Equilibrium will be reached at 70 millimeters of mercury. This drop in partial pressure of oxygen would result in less oxygen reaching the tissue and decrease the amount of oxygen available to meet metabolic demand. The third step of external respiration is the transportation of oxygen and carbon dioxide within the blood to the tissue. At a partial pressure of oxygen at 100 millimeters of mercury, we would only be able to transport 3 milliliters of oxygen per liter of blood if we were reliant strictly on the plasma. This is because oxygen is poorly diffused within plasma. At rest, the body's tissue demands 250 milliliters of oxygen per minute. If we are only able to transport 3 milliliters of oxygen per liter, we would have to pump 83 liters of blood through our pulmonary capillaries per minute. In order to meet the demand of the tissue and decrease the work of the heart, we need another source besides the plasma to transport oxygen to the tissue. This other method of transportation of oxygen is accomplished through hemoglobin, which are found in our red blood cells. Plasma carries approximately 1.5% of our dissolved oxygen in the blood. Hemoglobin carries 98.5% of our oxygen. Important to remember is that the partial pressure is measured by the diffused oxygen in the plasma. Any oxygen carried by the hemoglobin is not considered in reaching equilibrium between the alveoli and the pulmonary blood. The first step to diffusion is oxygen diffuses from the alveoli and into the capillaries. Each black dot within the capillary represents a molecule of oxygen. As the plasma comes into contact with a red blood cell, the oxygen is diffused into the red blood cell due to a concentration gradient of low oxygen within the red blood cell and higher oxygen within the plasma. As these oxygen molecules are brought into the red blood cell, they attach to the heme group of the hemoglobin and no longer contributes to the concentration gradient. As you can see, there is now a decrease in the plasma concentration of oxygen. This reestablishes the diffusion gradient and more oxygen is brought in from the alveoli into the plasma. The continuation of this process results in more oxygen being attached to the heme group within the hemoglobin and more oxygen being able to diffuse into the plasma of the blood. This allows for more and more oxygen to be diffused between the alveoli and the plasma. In this simplified example, let's say equilibrium was reached when 5 molecules of oxygen was diffused into the plasma. Without the presence of red blood cells, equilibrium would be reached once those 5 oxygen molecules reach the plasma. However, each red blood cell had 4 heme groups that could each take 4 molecules of oxygen. This means that each red blood cell is capable of holding on to 16 molecules of oxygen. Because we have 2 red blood cells, we were able to bring in 37 molecules of oxygen instead of the original 5 molecules of oxygen to reach equilibrium. This ability for red blood cells to hold on to oxygen molecules and not contribute to reaching equilibrium between the plasma and the alveoli is how our body is able to increase its oxygen carrying capacity. If we added a 3rd red blood cell, we can see how this would increase the carrying capacity of oxygen again. 16 more molecules of oxygen would move into the red blood cell from the plasma. Again, enough oxygen from the alveoli would move into the plasma to maintain those 5 molecules of oxygen to maintain equilibrium. Now we are carrying 48 molecules of oxygen plus the 5 molecules of oxygen in the plasma to maintain equilibrium and have increased our carrying capacity. We are able to exponentially increase our oxygen carrying capacity, which allows our heart to have to work less hard to meet the resting oxygen need of 250 milliliters of oxygen per minute. The red dashed line shows the hemoglobin saturation curve as it truly is, with a steep drop-off at low partial pressures and a long plateau at high partial pressures. The binding of hemoglobin and oxygen is a reversible reaction. This allows us to pick up oxygen in the pulmonary system and then release oxygen at the tissue level. The amount of oxygen the hemoglobin is able to pick up is dependent on the partial pressure of oxygen at that moment in time. The percent of hemoglobin saturation is a way to say how full is the hemoglobin. Is it 0% full, carrying no oxygen, or is it 100% full, meaning that every heme group within the red blood cell is attached to oxygen? This range between 0 and 100% saturation is determined by the partial pressure of oxygen. At the alveoli, the partial pressure of oxygen is 100 millimeters of mercury. At this level, hemoglobin saturation is 97.5% full. The average partial pressure of oxygen in the systemic system is 40 millimeters of mercury. At this partial pressure, hemoglobin saturation can only be 75%. This means once the hemoglobin reaches the tissue, it must release 22.5% of its oxygen that it is carrying. The hemoglobin saturation curve does not follow a linear trend. The importance of the plateau portion of the hemoglobin saturation curve ensures that our tissue receives adequate oxygen regardless of the partial pressure of oxygen. At normal conditions, our alveolar partial pressure of oxygen is 100 millimeters of mercury. This allows the hemoglobin to be 97.5% filled. What if all of a sudden we were dropped in Denver and the partial pressure of oxygen dropped to 90 millimeters of mercury because of the plateau portion, or the very flat line and slow decrease of hemoglobin saturation? Even if we all of a sudden had a 10% drop in partial pressure of oxygen, we would only see a 2.5% drop in hemoglobin saturation. This plateau portion of the curve ensures that we continue to have an adequate amount of oxygen available to our body. In fact, even if we were dropped, such as Everest Base Camp, and had a 40% reduction in partial pressure, all the way down to 60 millimeters of mercury, our hemoglobin would still be 90% saturated. The second goal of hemoglobin is to release the oxygen to the cells. At rest, the average partial pressure of oxygen in the systemic system is 40 millimeters of mercury. At 40 millimeters of mercury, the hemoglobin can only be 75% saturated. This means it has to release 22.5% of its oxygen to the tissue. What happens if our tissue becomes increasingly active? Because we are running, our tissue is using more oxygen, and it will therefore drop the partial pressure in that area. Here we see only a 10 millimeter drop of partial pressure of oxygen from 40 to 30. This same drop in the pulmonary system on the plateau portion of the curve resulted in only a 2.5% drop of oxygen saturation. However, here at the tissue level, on the steep end of the curve, we see that a 10 millimeter of mercury drop in partial pressure of oxygen causes the hemoglobin to have to release 50% of the oxygen bound to it. This increases the percent of oxygen released from the hemoglobin from 22.5% to 47.5%. This means a 10 millimeter of mercury drop on the steep portion of the graph is going to result in a doubling of the oxygen available to the tissue to use to produce ATP. Depending on the conditions around the cells, the curve can shift upward or downward. As an area becomes more concentrated with carbon dioxide, hydrogen, or experiences an increase in temperature, hemoglobin has a harder time holding on to oxygen. This means at the same partial pressure, it will release more oxygen than in normal conditions. An increase in carbon dioxide, an increase in hydrogen concentration, and an increase in temperature are all indicators more metabolically active tissue. Why would we want the curve to shift downward? If we had normal concentrations of carbon dioxide, hydrogen, and temperature, we would have a hemoglobin saturation of 75%. If the tissue becomes more metabolically active and sees an increase in carbon dioxide, hydrogen, and an increase in temp at the same partial pressure of oxygen, the hemoglobin will be forced to give up a larger percentage of oxygen to the tissue. So not only is metabolically active tissue going to have a lower partial pressure of oxygen due to the tissue bringing in more oxygen from the plasma, the hemoglobin will also have an increased dissociation from the oxygen and provide an excess of oxygen to the tissue. As the blood enters the capillaries, it has a partial pressure of oxygen of 100 millimeters of mercury and a partial pressure of carbon dioxide of 40 millimeters of mercury. The average pressure within the capillaries, however, drops to 40 millimeters of mercury partial pressure for oxygen and increases in partial pressure of carbon dioxide to 46 millimeters of mercury. The drop in the partial pressure of oxygen is going to cause the oxygen bound to the hemoglobin to diffuse off of the hemoglobin and into the plasma. And within the cells, where carbon dioxide partial pressure is greater than 46 millimeters of mercury, and the partial pressure of oxygen is less than 40 millimeters of mercury. The oxygen from the capillaries will diffuse from the capillaries into the interstitial fluid and into the cells. Carbon dioxide will diffuse out of the cells into the interstitial fluid and then dissolve into the capillaries, moving down its gradient from high to low partial pressure. Since the partial pressure of oxygen is only 40 millimeters of mercury, oxygen will dissociate from the hemoglobin and move into the plasma, where it then diffuses across the interstitial fluid and into the tissue. How do we transport carbon dioxide back to the lungs to be expired through our airways? Based on the partial pressures, we can see that oxygen will move from the blood into the tissue, and carbon dioxide will move from the tissue into the blood. Similar to oxygen, carbon dioxide is not readily dissolved within the plasma. Plasma can only hold about 10% of the total carbon dioxide that our tissues produce at rest. Blood coming into the capillaries is almost 100% saturated. It has a saturation of 97.5%. However, when it experiences a lower partial pressure of 40 millimeters of mercury that is found in the capillaries, it no longer can hold on to all of this oxygen. It has to release approximately 25% of the oxygen bound to the hemoglobin. The second red blood cell represents the red blood cell after it has disassociated from 25% of the oxygen it was carrying. The plasma can only hold on to approximately 10% of the carbon dioxide moved into the plasma. The remainder is taken care of by the red blood cells that have now disassociated from oxygen and have an increased carrying capacity for other molecules. Because the concentration of carbon dioxide within the red blood cell is low, carbon dioxide again diffuses down its concentration gradient from high concentration in the plasma to low concentration within the red blood cell. Once carbon dioxide enters into the red blood cell, it can have one of two fates. The first fate of carbon dioxide that diffuses directly into the red blood cell is it can combine with the hemoglobin and simply take the spot that was vacated by the oxygen. This again is a reversible reaction where hemoglobin can bind temporarily to carbon dioxide and later release carbon dioxide. About 30% of our carbon dioxide is directly bound to hemoglobin. The other 60% of our carbon dioxide is taken care of by our bicarbonate system. Carbon dioxide combines with water and through a reaction catalyzed by carbonic anhydrase react to form a hydrogen molecule and a bicarbonate molecule. The hydrogen molecule will then reversibly attach to the hemoglobin within the red blood cell, and the bicarbonate will dissolve out of the red blood cell back into the plasma. The ability for the hemoglobin to bind to hydrogen helps maintain normal pH within the blood. As tissue becomes more metabolically active, it increases the amount of carbon dioxide it produces and increases the amount of oxygen that it utilizes. The more oxygen a tissue uses, the larger the partial pressure of oxygen will fall within that tissue. Likewise, more metabolically active the tissue is, the more carbon dioxide it will produce, which will increase the gradient between the tissue and the capillaries. The decrease in partial pressure of oxygen causes more oxygen to disassociate from the hemoglobin to move into the tissue. The increasing gradient between carbon dioxide in the tissue and carbon dioxide in the capillary increases the speed of diffusion. Because more oxygen has left the red blood cells, there is now even more room for the red blood cells to carry carbon dioxide and hydrogen away from the tissue back to the lungs. When the blood returns back to the alveoli, we see a change in partial pressure where the alveoli have the partial pressure of oxygen of 100 millimeters of mercury compared to the capillary partial pressure of oxygen of 40. The partial pressure of carbon dioxide has reversed as well, where the alveoli is only 40 millimeters of mercury, where the capillaries are 46 millimeters of mercury. At this higher partial pressure, the hemoglobin wants to become fully saturated with oxygen again. That causes it to release the carbon dioxide, where it will dissolve into the plasma and out into the alveoli. The oxygen will then diffuse from the alveoli into the capillaries and bind onto the hemoglobin that the carbon dioxide has now vacated. The high partial pressure of oxygen also causes the hemoglobin to disassociate from the hydrogen ion that it is carrying. The bicarb moves back into the red blood cell and the reaction is reversed. The bicarb combines with the hydrogen and the reaction is catalyzed again by the enzyme carbonic anhydrase, and water and carbon dioxide are the result. This carbon dioxide is then moved back out of the red blood cell and is diffused into the alveoli. There is now an increase of space within the red blood cells to bind with oxygen. As the blood leaves the pulmonary capillaries, it again has a saturation of 97.5% oxygen. This blood then returns to the tissue and the process is repeated.